Hope you'll stick around and write down a few questions for us to challenge us. We haven't had enough interaction so far today. Okay, representational similarity analysis is a very simple way of looking at pattern representations measured with any technique of brain activity measurement. Could be fMRI, could be MEG, could be cell recordings. Who's familiar with pattern decoding techniques before the previous talk? Who's familiar with this? Okay, who's familiar with representational similarity analysis? All right, also quite a few. We'll go through this in a nutshell anyway, because there's a number of people here who haven't heard about it. So in representational similarity analysis, we present a number of stimuli, and usually we like to make each particular stimulus a separate condition in the experiment. And then for a given region of interest in our fMRI data set, we measure an activity pattern. So this could be a little ROI that you see there, and for a different stimulus, we have a different pattern of activity in that same ROI. So you see in the color code here the activity level. So this is the same region, but the colors are a little bit different, indicating the different patterns which we interpret as the representations of these two stimuli. And then in order to characterize the representational space of all the stimuli, we compare these patterns. So we measure in some way the representational dissimilarity, and this is what I'm going to talk about a little later in more detail, how to do that. One way to do it is by just correlating these patterns across the voxels, and then if we want, that would be a similarity. To make it a dissimilarity, we can take 1 minus that correlation. So once we've estimated that dissimilarity, we can enter it into a matrix. So here you have the little umbrella, and here the hand, and this will be one value here, the dissimilarity estimate. Now the colors here have a different meaning. They now refer to pattern dissimilarities rather than activity levels. And we can assemble all the comparisons between pairs of stimuli in this matrix, which we call the representational dissimilarity matrix. And this is useful because it allows us to compare the representational space in a brain region to representations obtained by other means. For example, it could be the representation in a simple stimulus description, such as the pixel-based dissimilarity. It could be a representation in a computational model that we show our particular images to. So the model would process the visual images, and then it's got its own internal representational patterns, and we can play the same game and look at the dissimilarity matrices for these representations. Or it could be a dissimilarity matrix obtained from behavioral data, for example, dissimilarity judgments, where we ask people to tell us how dissimilar each pair of stimuli is. Or it could be a task such as discrimination, where we could try to infer from reaction times how similar in the mental representation different pairs of stimuli are. So once we're in this format where we're comparing each stimulus to each other stimulus, we've abstracted from the voxels here. And this is very useful because it allows us to compare representational spaces, for example, between fMRI voxels and single unit recordings, or between fMRI voxels and the units of a computational model, or between fMRI and MEG, or between two different regions in the same person's brain, or between two different individuals. All of these comparisons are very difficult when you don't know how the voxels within the region of one person map onto the voxels in another person's brain. So we're kind of sidestepping this problem of defining the correspondency by just looking at the representational dissimilarities. So we call these matrices representational dissimilarity matrices. There have been quite a number of studies using this technique. Here's one where Mariko Moore predicted similarity judgments from brain representations. Here's another one where Winrich Freiwald looked at these matrices for measurements of monkey face patches, so cell recordings, and he found this beautiful transformation of the representation across these face patches. So in the middle face patch, everything clusters according to the view, and in the anterior face patch, you see this beautiful view-invariant representation of identities within each view. You have these little identities here. In 2013, we reviewed a number of studies. Since then, there have been many new studies. I just want to show you a couple that I'm particularly excited about. Here's one that used RSA on MEG to look in detail at the representational dynamics, so how representations unfold in time, millisecond to millisecond. So you have these representational dissimilarity matrices as a movie, and you can see how different categorical divisions come up in sequence. And this can tell us something about the dynamic process and recurrent processing in the brain. Here's another one from my lab by Jan Scharre, who found that the representations in inferior temporal cortex are unique to each individual, and my inferior temporal representational space will predict my perceived similarity of objects better than yours. So there's a subject-unique relationship between these representational spaces as reflected in our brains and our similarity judgments in this case. It's also not just perception. Here's a study that came out a couple of weeks ago from Jörn Dietrichson's lab, where they showed that hand use predicts the structure of representations of digits in sensory motor cortex. So they looked at the representations of the 5 digits of the hand in sensory motor cortex, and they had different models for predicting that representational space, and found that sort of the functional use model best predicted the structure. So this is just to share a bit of the excitement about what we can do with this technique. I want to focus on 2 questions today. One is how we can best measure the representational dissimilarities, and the second is how we can use these matrices of representational dissimilarities to test computational models. So on the first question, a basic point to appreciate is that distance estimates are positively biased. So imagine you have a representational space. Here I've sketched it out with these arrows indicating activities in different neurons. So imagine you have 2 true patterns, and you would like to estimate the distance in this representational space spanned by the neurons between these 2 patterns. Your data are always going to be affected by noise, so your estimate of pattern 1 from the data is going to be offset by noise in this representational space. And the same is going to be true for your estimate of pattern 2, and therefore the distance that you measure is going to tend to be larger than the true distance. Because in a multidimensional space, the noise is more likely to push 2 patterns apart from each other than to push them together. So these 2 cases are not equally probable, and that creates a bias in the distance estimate. So when we play around with MATLAB in a few lines, we can visualize this. So here you have the distance in the data. So these are the distances of the noisy pattern estimates. And here are the true distances before the noise was added. And you see that when the true distance is close to 0, then there's really this big bias, this big positive bias, where the measured distance is always much bigger than the true distance. Note that the distance measures such as the Euclidean distance are by definition positive. They can never be negative, right? So the way they are defined. And that in itself so clarifies that there's going to be a positive bias when there's noise in the data. So distances are positively biased, and that's something that they share with training set decoding accuracies. You've heard in Yuki's talk that it's key in pattern decoding analyses to cross-validate, right? So once you fit your classifier, it's not enough to look at how well that classifier does on your training data, because in a high-dimensional space, your classifier might do perfectly, even when there's no information at all about the labels in the data, right? So we need to cross-validate, and this positive bias at the distances is actually exactly the same effect that gives you a positive bias of training set decoding accuracies. And since RSA and decoding have been two popular approaches, it's worth looking a little bit more in detail at the relationship between the two. So here, let's recall the decoding scenario where we might have two conditions. Think of them as two particular stimuli. And here I've just drawn a voxel 1 activity and voxel 2 activity, but think of this as a higher dimensional representational space. And now we have these multiple estimates, so multiple repetitions of the same stimulus for condition 1 and for condition 2. And we could model these distributions as multi-normals, perhaps assuming that the covariance is the same between these two, and then we'd get an estimate of the optimal decision boundary. So we could estimate the Fisher linear discriminant, which is a vector orthogonal to that optimal decision boundary. Now, recall that it's not enough to just look at the separation of these two clouds of points on this discriminant because of the bias, because of the overfitting of the discriminant to this data set. So what we usually do in pattern decoding analyses is we take a separate data set, I've called it run B here, and in the separate data set we've got replications of these measurements for these stimuli. And now we could play the same game here, but instead we take the Fisher linear discriminant from run A and apply that to the run B data. And then we can drop these points, the data points from run B, onto the Fisher linear discriminant from run A. And now we're in a position to do inference and assess whether there's really a difference between these two conditions in the representation. So, but this is interesting. Once we've dropped these points onto the discriminant, we're in this situation. We have essentially univariate data, and we have all these red points, we have these blue points, and we want to know, we have the hypothesis that the blue points are further toward this end of the dimension, and the red points are further on that side. Now, in decoding, what we do is we use this threshold, and then we count how many of these points are on the expected side of this boundary, and that's the decoding accuracy, and then we do inferential statistics on that. So that's our test statistic, and then we want to determine whether that's greater than expected by chance. However, what we have here is a really very simple univariate scenario where we might also just do a t-test, right? Actually, the t-test is a very good thing to do in this situation because the data already are close to multivariate normal to begin with. We always assume univariate normality in univariate fMRI analyses. And now we have, after projecting onto this Fisher linear discriminant, we have a linear combination of lots of things that are already close to normal. So the assumptions of the t-test are actually very not violated here at all. It's a very good thing to do. So this suggests using the linear discriminant contrast, LDC, or the linear discriminant t-value, which is this contrast normalized by its standard error, as a measure of the separation in the multivariate space of two patterns. So it's a really very straightforward way of estimating the separation. However, note that this is a continuous measure, so it's more like a distance in that it's continuous. It's not quantized like an accuracy, and it doesn't saturate like an accuracy. An accuracy, when it's 100%, cannot get any larger when the distance in the space gets larger. So it has some desirable properties for our purposes of characterizing the representational geometry. But it's also cross-validated, much like decoding accuracies, and therefore unbiased. So if you're familiar with the Mahalanobis distance, this is a slide for you. The definition of the Mahalanobis distance is this: pattern 2 minus pattern 1 transpose times the inverse of the noise covariance matrix multiplied by the difference between the two patterns. So this is just a definition of the Mahalanobis distance. Now note how related that is to the Fisher linear discriminant contrast, where this is the Fisher linear discriminant vector W, and this is the difference between the two patterns in the test set. So the Fisher linear discriminant contrast is really exactly the same thing as the Mahalanobis distance, only that we swap out the data sets for the two pattern estimates on the right-hand side of the equation. So when we do that, then we get an estimate of the distance that's multivariately normalized, like the Mahalanobis distance, but that is also unbiased and would be symmetrically distributed about 0 if the true distance were 0. So here's a demonstration on that from a simple simulation in an upcoming paper by my graduate student Alexander Walter. So here the true distance is in different shades of gray here. When the noise level is essentially 0, you just estimate the true distance correctly, even without cross-validation. But then as the noise goes up, you get this bias, and all the distance estimates go up. When you use a cross-validated distance instead, you estimate the y-axis is scaled differently here. All the distances are now accurately estimated, and as the noise goes up, the variability gets greater, but the mean value stays the same. So we've looked at the reliability of different dissimilarity measures in terms of the RDM split half correlation. So when we just take two data sets, we estimate the RDM for the same conditions. This is a replication of the experiment. Of course, we would like the two RDMs to be as similar as possible, and we'd prefer measures of dissimilarity that ensure this. And when we do these comparisons, we see that the best performing distances are those that use multivariate noise normalization. So that includes all of these different distances applied to the data after whitening, spatially whitening the pattern space. So here we have in gray and dark gray the Euclidean and Mahalanobis distance and the correlation distance in a lighter shade. Then in blue, we have decoding accuracies interpreted as distances here. When the accuracy is larger, we have a greater separation of the patterns in the pattern space, and we just use that as though it was a distance. And here we have the continuous cross-validated distances, LDC and LDT. And what we saw in general was that the multivariately normalized distances performed much better than the univariately or unnormalized distances, and also that these continuous cross-validated distances were particularly reliable. So this is just one particular data set. We tried this out with 4 different data sets and also looked at a number of different measures of reliability. Here it's the Pearson correlation between the RDMs. Also did it for the Spearman correlation, for the Pearson correlation with the 0 point fixed, because now we have unbiased distances, so we can actually interpret the 0 point, so it makes sense to look at that. And also for the coefficient of determination. So when we did that for all of these scenarios, the story was sort of consistent with the summary that I just gave you. So we're quite confident that these results generally hold up, and we're in favor, we tend to favor these continuous cross-validated distances now. We also tried these different measures of multivariate separation in decoding scenarios, because we argued, well, if they're actually sort of more sensitive, if they give us more precise estimates, then they should also provide for more sensitive decoding of pairs of stimuli. So what we did was we looked at a data set where we had lots of different objects and lots of different categories, and then we tried to decode every pair of categories within each of a large number of different categories. We tried to decode every pair of exemplars, and we did this for each of a number of different brain regions. So we had lots of different scenarios, some of them easy decoding tasks, some of them hard decoding tasks, to test sort of how well do different measures work. First, we tested that all our different approaches to testing for significant pattern discriminability were statistically valid. So we used simulations to test these measures, and we found that all these different approaches that we tested met the nominal 5% false positives rate. So all these dissimilarity measures can be used to test for pattern discriminability. But then when we looked at how well they perform, in how many cases they detect differences, we find that the number of significant cases detected is very different. And for the Mahalanobis distance and the LDT distance, there's a really much larger proportion of cases where we have positive findings. So we conclude from this that also in practice, also when you're not interested in estimating the distance, but you're just asking the simpler decoding question, is the distance greater than 0? It's good to use these distances that use multivariate noise normalization. So putting these points together, we have different ways of measuring representational distances. Some of them are without cross-validation. These include the Euclidean distance, the Mahalanobis distance, and the training set decoding accuracy. We also have cross-validated variants of these. For the Euclidean distance, that would be the centroid connection discriminant contrast. For the Mahalanobis distance, it would be the Fisher linear discriminant contrast. And for decoding, it would be the test set decoding accuracies. We don't like these uncross-validated ones so much because they're positively biased, making it impossible to interpret the 0 point. These ones on top are blind to the covariance, so that's kind of a drawback and makes them less sensitive also. And these decoding accuracies are ceiling limited because they can't exceed 100%, and they're quantized, especially when there are not so many different levels to the possible decoding accuracies, and that makes them less reliable. So in this picture, the Fisher linear discriminant contrast starts looking very good, and this is sort of what we're using now to measure representational distances. So to summarize this part, continuous distances with multivariate noise normalization provide the most reliable estimates. Classifier accuracies are not optimally reliable, and cross-validated distances, LDC and LDT, are unbiased and have an interpretable 0 point and are therefore attractive. Okay, so let's go to the second question. How can we test computational models? We were interested in testing a deep convolutional neural network, and this is the kind of neural network that's currently revolutionizing artificial intelligence and starting with computer vision. So this is the kind of system that's now the state of the art in computer vision. It's been trained with stochastic gradient descent, supervised with 1.2 million category labeled images, and it's got 60 million parameters and 650,000 neurons. We were interested in asking whether this network is functionally similar to the brain, and more specifically, whether these layers of representation in this network can explain the representations along the ventral stream. I'm going to focus on inferior temporal cortex because that's sort of the highest point, the apex of the ventral stream that we really want to understand, and that's been very difficult to computationally tackle so far. So how do we approach this with RSA? So what I'm plotting here is the accuracy of the human IT dissimilarity matrix prediction. So that's the correlation between the RDM, the representational dissimilarity matrix, predicted by a layer of the model. So we just presented the stimuli to the model and then measured the dissimilarities in each of the representations of the model. And the corresponding RDM for IT. And the way we measure this is by Kandel's tau A, and that's just a correlation coefficient. You can ask me in person why we use this particular one. We could also use the Pearson correlation or the Spearman correlation with slightly different implications. And what I'm plotting here is the group average, so across our group of subjects, of the Kandel's tau A. So for layer 1 of the model, we get this very small bar here. So this is kind of disappointing. If you thought that layer 1 would explain inferior temporal cortex well, you'd be quite disappointed here. However, the correlation is significant between the RDM of layer 1 and IT. And this indicates that if we used layer 1 as a decoding model, then we'd get significant decoding. So we can already conclude from this that the region at least contains some information about the stimuli, right? So my point here is the scenario we're looking at here is one in which the model is actually interesting in that it is a computational model that mimics, or that we would like to mimic, what the brain does. But if we evaluate even a low level of this model by the criterion of decoding, then we can find sort of a significant relationship between stimuli and responses. However, that's a very low bar. You can also get an error bar on this level of relationship between these two representational dissimilarity matrices by bootstrapping the stimulus set. But the important point is that this significant effect is small relative to the effect that we would expect the true model to achieve. So here's what we call the noise ceiling, where the upper bound of the noise ceiling is the highest accuracy that any model can achieve given the noise and the variability between subjects in the data. And the lower bound of the noise ceiling is the other subject's average as model compared to each subject's RDM. So that's using the other subjects as a stand-in for the true model to estimate how well the true model might be doing. And then because we have limited data, the other subject's average is a noisy estimate. It's not a perfect estimate, and therefore it provides a lower bound on the performance that we would expect the true model to have. So we have the expectation that the true model would fall into the noise ceiling range here. So as we go up the layers of the model, we explain more and more variance in this representation. Layer 3, layer 4, layer 5. These are the convolutional layers of this network. Layer 6 and layer 7 get quite close to the noise ceiling, but don't quite reach it. And then when we fit SVM discriminants on separate sets of images to emphasize the right categorical divisions that are prominent in inferior temporal cortex, then we reach into the noise ceiling. We also fitted a weighted combination of the fully connected layers and the SVM discriminants and reached even deeper into the noise ceiling here. Now, it's nice to know that representations in computational models explain significant variance, it's even better to see that they perform about as well as any model can perform given the noise in the data. But it's also important to ask whether these different model representations are significantly different. And a simple way to assess that is by bootstrap resampling the stimulus set. So we just say our set of stimuli are somewhat random, of course, always, right? When we design our experiments, we don't think of our stimulus set as a very definite decision defining what we're interested in. We're interested in something more general, and then we use a set of stimuli to study that. But really, we would like to be able to generalize from our particular stimuli to a whole population of stimuli, ideally. And one way of studying the effect of the stimulus set on the results is by bootstrap resampling the stimuli. So if you have a rich stimulus set, then you can do that, and you can look at the variability of all the effects you're measuring. And this is a way to compare different models here. So when you see a connecting line, that means that this bar is significantly higher than this bar here. So lots of these pairwise comparisons are significant. Most of them are, but not all of them. For example, these two models are not significantly different. So this is a way for us to assess whether two models are actually significantly different. So what do we learn from this? We have a model here that reaches the noise ceiling. What does that tell us? Is it the true model? Probably not, right? It means that our data are not good enough to distinguish this model from the true model, right? So the fact that we're hitting the noise ceiling tells us that we should go and get better data, which will push up the noise ceiling and reveal whether this model is actually true, or whether this model still holds up, or whether we need to consider other models. So in conclusion, insights on testing computational models. We can go beyond decoding stimulus information and test explicit computational models of brain information processing. Representations in brains and models can be characterized by representational dissimilarity matrices. Multiple models can be statistically compared by simple non-parametric statistical inference. And the noise ceiling tells us whether a model fully explains our data, guiding us to seek either a better model or more data. Thank you. We might actually have time for one question. Or we can do the discussion after Jack's talk. Maybe that's fun too. All right, if there's no question,